All right, so I'm going to tell you a little bit about the Developmental Synaptopathies Consortium, and particularly the Syngap piece of that. Here are my disclosures. So the concept of the Developmental Synaptopathies Consortium is that many neurogenetic disorders, as you've already heard today, are associated with autism and intellectual disability, and those disorders converge on a few common pathways. And so that leaves us opportunities not only to learn about the distinctions and differences between those disorders, but also about the shared pathophysiology. And because of that shared pathophysiology, there are shared possibilities for treatments, shared phenotypes, and shared opportunities to develop biomarkers for outcome monitoring in clinical trials. So I want to give you a little bit of history of the Developmental Synaptopathies Consortium. So this has been going on for about 10 years now, and we are now in years 11 to 15. In the first 10 years, we were focused primarily on synaptopathies under the umbrella of mTOR pathway disorders, so tuberous sclerosis, Phelan-McDermid, and P10. Syngap is coming, so hang on for one slide. This was funded by an NIH U54. Mustafa Sahin is the PI. We were focusing primarily on the natural history, developing some biomarkers, and there already have been a couple of spin-off clinical trials as a result of that first 10 years of work. Thanks to the support of Cure SYNGAP1, I did it. We piloted a multi-site study of EEG biomarkers in Syngap using the same paradigms that we had used for the rest of the Developmental Synaptopathies Consortium, starting in 2021. And so far, that work has led us to develop clinical outcome assessments that are being used in clinical trials for those other disorders. We've developed biomarkers for use in clinical trials. We initiated 2 clinical treatment trials, one in tuberous sclerosis and one in P10. Collected some longitudinal natural history data, some of which led to FDA approval in Phelan-McDermid syndrome for the first in-human gene therapy trial. Consensus guidelines for those physicians for assessment and treatment, and also a bunch of training and mentoring of junior investigators along the way. So we're now in the years 11 to 15. We're just getting started. And the big addition is that we added Syngap to the Developmental Synaptopathies Consortium. We're continuing to study those other mTOR pathway disorders that we discussed. We're focusing a lot on sleep and sensory processing, and so I'm going to spend a little bit of time today telling you why we chose those 2 areas of focus, but we are still continuing to characterize cognition and communication and neuropsychiatric symptoms across the lifespan, including starting to look a little bit at catatonia in adolescence. And we're continuing to develop neurophysiological biomarkers, which I'll tell you a little bit about as well. So why are we focusing on sleep and sensory processing? So 1, these things are key aspects of functioning that really, really impact quality of life. We all know that if we don't sleep well, how we behave the next day, right? And sensory processing, we've heard a lot already about behavioral challenges, and we think that sensory processing differences are a huge, huge, huge contributor to those behavioral challenges, but they are more tangible to develop biomarkers for. So we have opportunities from sleep and sensory processing for objective and brain-based biomarkers, which, as we know, are incredibly important for outcome measures for clinical trials. And sleep and sensory processing have not been extensively studied in any of these disorders, including Syngap. Both sleep and sleep and sensory processing provide these really kind of basic, low-level, accessible, measurable, translatable across species, across ages, pathways, and give us information, sort of windows into those higher level cognitive functions. And that's both because the pathways, the structural pathways for sleep and sensory processing are actually quite well mapped. We really understand exactly where the neurons and synapses are to process a stimulus from a touch or from a sound or from light into the eyes. We also know a lot about the computational framework, so how are the excitatory and different types of inhibitory interneurons interacting to adjust sleep and sensory processing. When we talk about sensory processing, I want to clarify, we are not only talking about the peripheral organs, right? We heard earlier today, well, children with Syngap are not deaf, right? But sensory processing, only a small piece of that is those peripheral organs, the eyes, the ears, the touch receptors, and the skin. After that signal comes into our bodies, a huge part of the nervous system's role is volume control. We decide what are we turning up and what are we turning down, right? If a sound is too loud, we turn it down. If a sound is not important, the refrigerator's humming, our nervous system turns it down. If a sound is quiet but important, like someone's whispering gossip about us at the table next to us, we turn it up, right? And that is a huge part of what the nervous system does. And we have some early data to suggest that in Syngap and in many of these other neurodevelopmental and neurogenetic disorders, that volume control is not working in the same way. And again, we think that's a big part of the reason why we're seeing a lot of these behavioral challenges. There was a nice study that came from Jimmy Holder's lab. Ariel Lyons-Warren was the first author. And what you can see here over on the left is that green box is typical responses, and the lighter blue is responses in Syngap. I want to highlight three things. One, almost everybody with Syngap has atypical sensory processing in almost every single type of sensory processing there is. Two, there's a paper from Kim Wiltrout, who has had funding from Cure Syngap, with Ann Peduri as the senior author, looking at medical record review of sensory processing challenges, which show that 22% of children with Syngap have reported sensory processing challenges. I don't think this is an ascertainment bias thing. I think we as clinicians are not asking about it. And so I think what we learned from this is that it's something that we really need to know more about and ask about. The third thing I want to highlight is that some of the questions we ask on here are opposites, right? So there's an entire box at the top here on the left called sensory seeking. And then the next one below it is sensory avoiding. Those are opposites. Almost nobody with Syngap has normal sensory seeking or normal sensory avoiding. So they're doing both. How is that possible? We've actually found this in idiopathic autism as well, and in fact, even in typical development. The people who have the greatest amount of sensory hyper-responsivity also have the greatest amount of sensory hypo-responsivity. We call that the sensory paradox, and I think that gives us some clues for really understanding sensory processing. So as we think about why this might happen, what we start to think about is intrinsic factors, like what is the brain doing left to its own devices on a moment-to-moment basis that might lead to these very brief periods of over-responsiveness followed by very brief periods of under-responsiveness and vice versa. Also, we want to think about extrinsic factors, characteristics of the stimulus, like how loud is a sound or how strong is a touch or how bright is the light. The temporal context, have we seen, heard, touched this before? And spatial context, which is sort of the cocktail party effect of what else is happening at the same time when a given sensory stimulus comes in. So I'm not going to tell you about all of these results because I did that, I think, a couple of years ago. But I do want to tell you how some of these thoughts are leading to our next steps in those years 11 to 15. This is some data, again, from idiopathic autism, looking at loudness. Essentially what we see in autism is that children with autism and adults actually over-respond to loud sounds, but under-respond to quiet sounds. When we look on EEG, what we see is that in the P1 component, this early positive going component that essentially detects the incoming information, everybody with autism and typical development is responding essentially in the same way and the expected way, which is that the stronger the stimulus, the louder the sound, the stronger the brain response. We expect that. What's interesting is the plot on the bottom right. So for that return negative going component, where the brain is essentially responding to that incoming stimulus and deciding what it's going to do with it, as the stimulus gets stronger, that negative component gets more negative. It gets stronger in pink, in typical development. So the brain sends a signal back that says, hey, that was really strong, FYI, to the thalamus, right? Or, hey, that wasn't very strong. And then the brain can predict or decide what to do next. In autism, that feedback is not happening at the same, in any manner that's associated with the strength of the incoming signal. The brain is just going, yep, got a signal, and it's not adjusting its responses in the expected way. And we think that's part of the reason why we're getting this over and under responsiveness. We see the same thing with habituation. If you just repeat a sound a few times, in typical development in pink, the brain's response on EEG will go down over time. In autism, in many children, we don't see that. We actually see that the brain facilitates to this repeated response. The brain's response goes up over time. And again, you see this sort of phenomenon of the crossed lines, right? Where if you kind of extend these out, early on, the brain in autism is under-responding in the brain to the first stimulus, but then over-responding to the last one, right? And so we think we're going to see something in Syngap, and there is some early data from 8 participants with Syngap showing that, in fact, this is very true for habituation, that if you look on the left, the typically developing controls have the expected habituation response, and in Syngap, that response is sort of all over the map. Many of them are not habituating in the expected way. Sleep. We're hearing numbers ranging from 60 to 62%, this is a very tight range, of children with Syngap have reported sleep problems. Now again, we don't know what yet, we're about to hear a little bit more, I think, what's happening in the brain during sleep, but from what we can tell, we see sleep problems in 62% of these children. Those sleep problems, like we said earlier, are associated with things like anxiety, depression. It interacts with those sensory sensitivities and behavioral problems, right? So understanding sleep can really give us a window into those things as well. And improving sleep can improve those things. So here's how we're setting up the study. We're just getting started for this third round. So 4 synaptopathies now, those 3 mTOR pathway disorders and Syngap. We're going to do neuropsych assessments on 100 individuals with Syngap, age over 18 months. That includes sensory measurements on neuropsych testing, sleep measurements on neuropsych testing, and we also have a typically developing control group that will have a shortened neuropsych battery. We're also collecting electrophysiological biomarkers on 50 of these individuals. These are children aged 2 to 11. And we're going to collect more targeted sensory EEG than we did last round based on, this is really hypothesis-driven work based on what I just told you about. So we're going to collect resting data still, but also some targeted paradigms looking at response to loudness and some habituation paradigms as well, because we think those are really fruitful opportunities for potential biomarkers for clinical trials. We're also going to collect sleep EEG. That's going to be collected in the home environment for 7 days. Right now the plan is to use Muse, but we haven't bought the headbands yet, and I know we're about to hear from Beacon in like 5 minutes, and actigraphy as well. Here are our sites. This is a six-site network. You will note that there is some overlap with, particularly with Promise that we heard. We think that's a good thing. There are a lot of opportunities here, I think, for collaboration and for crosstalk. There is a huge, I get to represent this group today, but there is a huge group of people, really smart people, doing this work. We're divided up both at the bottom, you can see by the clinical research studies by the neurogenetic disorder. And we have a whole subset of different cores: administrative, biomarkers, neuropsych, biostats. We have a steering committee. We're working very closely with the patient advocacy groups. We also have some pilot studies and a pilot feasibility core, as well as a career enhancement core. So I think there are a lot of good opportunities that will come out of this. We are looking forward to getting started, and we are also open to any suggestions or thoughts that you might have as we get started. So I'll stop there and say thank you.